Okay, welcome back, grade 12s. So what we're going to go through today is kind of the purpose of DNA. So the purpose of DNA, we went through last week about replication and how important it is to ensure that you replicate everything perfectly and that you have that code, every single time you copy that code, it's perfect. Well, why? Why does that code need to be so perfect? Well, the code codes for something, okay? And what it codes for is protein. And that's what we're going to go through today is how exactly that happens. There's going to be two parts to this lecture. Now, as you go through it, again, we're getting to kind of a complicated area, and I'm struggling to kind of, as we have this limited course material, trying to limit the amount of detail that you guys need to absorb, but still covering the concepts. So, it might be a little bit difficult the first time through as we go through it, but, as we go through a couple more videos I'm going to post tomorrow, hopefully it becomes a little bit clearer. So this is the general idea. When you want to make a protein, you have to take your DNA, okay? And what you have to do is you have to take half of it. So you either take the bottom or the top of the double helix, okay? So in this case, they took the bottom of the double helix, okay? And then what they did was they made a copy of it. Now, when they copied it, you notice it's complementary. It's just like replication. So this is the DNA code, and what they're going to do is they're going to make a one-stranded copy of just that section of DNA, not the entire DNA, just that section, which we call a gene. Okay? So it goes from DNA to what we call single-stranded, and now it's mRNA. So this is now the code. Then what happens is we then read this code, and it turns into what we ultimately want is a polypeptide or a protein. Okay? Now, this is a very simplistic overview of it, so now let's kind of go through the bigger. Okay, so to understand the big picture of protein synthesis, it's handy to think of DNA, or the entire double helix, or 23 chromosomes, as a recipe book for protein. Okay, so very similar to how you have, a recipe book over here, and inside your recipe book you have all kinds of different recipes, one of them being for cookies. DNA is the exact same thing. This is a recipe book, and it makes, instead of making different bake items, it makes protein. Okay. So that's kind of the start, as you kind of have to think of DNA as this huge book of recipes, of all the different things that you require for your body, both to build it and to run it. Okay. Now, to understand the big picture of protein synthesis, just like I already mentioned, it's handy to think of DNA as a book. Now, similar to how a recipe book has different chapters to organize the recipes in DNA, so like a recipe book might have something like, chapter 7, that's where all my cookie recipes are. While DNA, being a recipe book, has something very similar, but instead of having chapters, okay, what they have is chromosomes. So instead of saying like chapter 7, it would be chromosome 7, okay, which is kind of an area where all very similar recipes are together. So that might be, instead of like in the recipe book, it's for cookie recipes, but say in your chromosomes, on chromosome 7, it's all about facial features. Okay, so it's all kinds of different recipes for creating, your mouth, your nose, your eyes, your lips, and all the different parts of your face. Now, we can go one step further. Within, say, a recipe book, you're going to have a recipe book of cooking, and then you're going to have, like, say, chapter 7, okay? And then you're going to have recipe number 7 of chapter 7, which is specific, like a sugar cookie, okay? Well, the same thing with DNA. You're going to have DNA, but you're going to have chromosome 7, okay? But then on that chromosome 7, again, chromosome 7 is all the different facial features, you're going to have specific recipes, like instead of sugar cookie recipe, we're going to go with gene 7, the eye color recipe. Right? So the basic idea I want you to come from this is the entire DNA is the entire cookbook. Chromosomes are like individual chapters, and genes are like individual recipes from that. Okay, that's the easiest way of kind of about DNA. Now, let's go through an example, okay, of how this kind of really works. So take a look at a hair cell. So say you're a hair cell, and your job is to create color pigment. So if you look down here, kind of what I have highlighted here, this is a hair cell. Okay, so it's just a cell. It's an odd-looking cell. Okay, where it's not like a circle like we normally draw cells. It's a cell. So here's its body, and then it's got the long part of the hair growing out, and it's got your nucleus like every other cell would have. So there's our hair cell. Now inside that hair cell, you get instructions on how. You get an entire book of the DNA recipe book inside that nucleus. Okay? So each cell gets a whole copy of the book, so you have way more instructions that you're needing. Inside that hair cell, it gets the instructions for how to make a heart, how to make a brain, how to make teeth, how to grow bones. It doesn't need any of those instructions. It only needs instructions for how to make the brown pigment in color, but it has all of the instructions. So every cell gets a complete recipe book. Now, being the hair cell, you need to find the right chapter. There's a whole bunch of chapters that you're never going to read because it's not related to you. But you need to find the right chapter that's related to hair. So that's when you find the right chromosome. Right? And these are just some examples of what chromosomes look like. So let's say you're like, okay, this is the chromosome right here that's related to hair color. Okay? Now, on that chromosome, there's going to be a gene. That's the exact recipe. Okay? So that's the recipe now for the hair color. And this is what a recipe would look like. It would just be a whole bunch of series of letters, A, T, G's, and C's in that order. If you have brown hair, then you have the exact same recipe as every other person who has brown hair. If you have, say, blonde hair, then you might have 2 letters down here that are a little bit different. If you have red hair, you might have a couple letters here that are different. Most of the letters would be the same, but generally just a slight differences. Okay. So now, once you have that code, okay, and that code came from your DNA, right, then what you do, you're going to make a photocopy of what this recipe is. Okay? Now, this photocopy is called RNA, or specifically mRNA. So we're not going to actually take the actual DNA because the DNA is too valuable. What we do is we make a temporary copy. So if you can see down here, okay, where I'm highlighting, this is the mRNA copy. Okay? And the only difference is it's going to be basically, instead of having T's, it's going to have U's in the replacement. Okay? So it takes a copy of the DNA recipe. Now, once you have this mRNA, you bring it over to what we call a ribosome. The ribosome reads the mRNA, and then develops the polypeptide chain that relates to it. Okay? And that's a very, very simple, high-level overview of it. But now we're going to have to go through it, obviously, in a little more detail, because there's some gaps there. So we're going to break these 2 lectures into 2 pieces. The one lecture is going to be called transcription, okay? And that's what today's lecture is. And the other one that I'll do next is called translation. Now, the purpose of transcription, you can kind of see in the word right here, the root word in here is transcript. Okay? What you're going to be basically doing is creating a transcript from the DNA. Not the original copy of DNA, but you're going to create basically a temporary piece of it called mRNA. And that's the whole process of transcript, is we have to open up the DNA, photocopy a small section of it, and then we have our transcript. So that's what today's lecture is going to be on, is how does that happen. Okay, so let's go through this. So transcription is the first of 2 processes in protein synthesis, and protein synthesis just means making of protein. So transcription, as the name implies, is the process of creating a disposable transcript of code or instructions in the form of mRNA, okay, from the original DNA code to create a protein. So here's their original DNA code, okay, and what transcription is the making of this, okay. It's a single RNA, we call it mRNA, it's single-stranded, and it's basically just a small section of DNA, okay, which we call a gene. Okay? And then once we have this code, the next step, which we're going to cover on the next lecture, is turning it into translation. Okay, so transcription, the parts. So the first thing you're going to need to do is you need to locate the gene. Okay, so you have to locate within the DNA, well, where is the part that I'm looking for? Where is the code that I'm looking for? Okay, and in this case, on this example, the code they're looking for is right here. That's the gene. All those other letters that I don't have highlighted are not part of it. Okay, but that was the code right here. Actually, we're going to go a little bit farther. We're going to go all the way down to the bottom. Okay. So that's the gene. That's the gene for, say, hair color or for whatever protein that you're going to make. That's the recipe. So what we need to do is we need to get a copy of this. Okay? So the DNA opens up. So the first thing we do is we locate the gene. We've just done that. It's right there. Second step is to temporarily open up the double helix and make a disposable temporary copy of the gene in the form of pre-mRNA. Okay. Now, notice that it uses complementary base pairing. And what I mean by that is this is the gene down here that I've highlighted in red. Notice that the RNA or the mRNA is not the exact same letters, but complementary to it. So if the DNA says T, then the mRNA is A. Okay? If the DNA says A, then the mRNA says U, because there is no T in RNA. Okay? So it's complementary. So what this thing is going to do is we're just going to make a copy. We're going to go all the way down this little piece of DNA, and we're going to make a copy, and this is what we have. And this is the whole process. Okay? So we now have this, what we call pre-mRNA. Okay? So this is very close to being ready to be coded, but we have to go through one more step. Really, this is going to be way more letters than this. It's really going to be thousands and thousands of letters long. We've made a very, very short segment. But say this is 1,000 letters. Let's say, for example, that this little piece is 1,000 letters long. Okay? Not every single section in that 1,000 letters is required. There is some little pieces that we call non-coding regions. They're just in there. We don't exactly know why they're in there, but we know that they're not required. They're not part of the code. So before we send that, so the next step is the pre-mRNA needs to be modified. So we need to cut out within this area that we made, within all of these letters. So if I go back a letter, message to get coded, we have to cut out all those non-essential parts. And we call those non-essential parts introns. Okay, so here's a little part right here. I'm going to just set a better color, like say yellow. So here's an intron here, an intron here. And this little coding areas are areas that basically have no information. They don't have they need to be cut out. They're non-coding regions. The axons, okay, which we have that I'm highlighting here in blue, are the most important parts. We need to make sure that they actually get their left. So what we do is we cut out those introns. They leave, and you can see the introns. They get cut out, and they're right here. And then we have one nice solid piece that's all coding. And we call that mature mRNA. Okay? Now the last thing that we do is we also put this thing called a tail and a cap on it, and that's just to protect it from breaking down. Okay, so now we have our transcript or our code, okay? And now we're ready to now go to the stage. Okay, so going back here, we've now gone through transcription. So we're at this stage right now. We've got a transcript, okay, and the next lecture that we're going to go through is, well, how does this transcript then become a protein in the process called translation?